Hello, my name is Viktor Stolc. I'm a senior scientist at the NASA Ames Research Center. Thank you for the invitation to present our research results and the opportunity to explore some novel ideas about a potential connection between somatic mutation and metabolic roots of depression. This presentation is based on a long-standing collaboration with colleagues from NASA, Stanford School of Medicine, and SETI. We have been working on the problem of understanding timekeeping in biological systems, using genome science technology, high-performance computing, and next-generation sequencing in the unconventional and extreme environment of spaceflight, which induces metabolic stress in astronauts and in all studied model systems. It is now recognized that there is a need for fundamental study of somatic mutation because of the many varied phenotypes and genomes in all human diseases. The results and novel concepts I will present here are somewhat surprising and even have implications for the topic of this symposium, which is entitled How Studying Anxiety, Irritability, and Anhedonia May Improve the Knowledge of Depression. I hope you may find it interesting. I will make one overarching scientific comment which is relevant to my entire talk, and that is that time is an essential non-random variable of information content and processing functions in cellular systems. Please keep this in mind as you are listening to my presentation. Depression is a common and serious medical illness that affects how you feel, the way you think, and how you act. It can lead to a variety of emotional and physical problems and can decrease your ability to function at work and at home. Depression remains a multifactorial condition, influenced by various biological, psychological, and environmental factors. There is a growing body of research suggesting a link between the immune system and depression, which is a topic I won't be discussing here, but it certainly fits any discussion of novel concepts about depression. Lastly, I want to emphasize that depression is a complex condition influenced by various factors, including genetics and environmental stressors, which I suspect even experts in depression would agree with. Now, I would like my physician colleague, Dr. David Loftus, to present a case study which I think will be very instructive about how important it is to think of novel mechanisms of depression.
Thank you, Viktor. Well, I'm happy to present this case history to you, and I think you will appreciate it as well as the surprise ending. A 68-year-old physician informally sought the advice of a physician colleague of his, a neurologist, at a major academic medical center. The patient complained of 16 weeks of anhedonia, decline in performance at work, and easy fatigability. As a result of his symptoms, the patient was forced to scale back his clinical activities at the medical center. As reported by the patient's family complained that the patient's memory was failing. He had difficulty remembering recent conversations, and he missed several significant family events he had promised to attend. On one occasion, he misplaced his car. The patient reported insomnia, which he attributed to lack of physical exercise. Over the course of the next few months, the patient sought the advice of several additional physician colleagues at his place of employment, including another neurologist, an internist, and a psychiatrist. At the urging of the patient's psychiatrist colleague, the patient was started on an SSRI, sertraline. The patient did not report any benefit from taking sertraline. In fact, over the course of the next several weeks, the patient's symptoms worsened. The patient self-discontinued sertraline. About 8 months after the patient's depression began, the patient underwent a routine surgical procedure unrelated to his depression. Pre-op labs for the surgical procedure included coagulation studies, an electrolyte panel, and a complete blood count. The electrolyte panel and the coagulation studies were normal, but the CBC showed mild anemia with an MCV of 110, triggering a review of the blood smear by the hematopathologist. The blood smear showed macrocytic red blood cells, hypersegmented neutrophils, normal-appearing platelets, and normal-appearing lymphocytes. Reflex testing initiated by the clinical laboratory included a methylmalonic acid level that was markedly elevated, a vitamin B12 level that was slightly below normal range. Serum folate and plasma homocysteine levels were normal. A blood test for intrinsic factor antibody was strongly positive. The patient was started on vitamin B12 injections per an internist whom the patient reluctantly agreed would serve as his primary care provider. Within 2 weeks, the patient's depression and fatigue dramatically resolved, and he was able to return to a normal clinical schedule. Back to you, Viktor.
Thank you, David, for that compelling case study which illustrates a link between depression and essential nutrient deficiencies that can inspire us to explore novel ideas about the metabolic basis of depression. We now appreciate that when vitamin B12 deficiency exists, many undesirable processes involving reactive oxygen species develop in the body, including in the central nervous system, that can lead to depression. So for the rest of my presentation, I would like to discuss the causal links. And lastly, I would like to touch on the topic of nutraceuticals that may be used for treatment of depression. To begin to understand the biochemical link between depression and essential nutrient deficiencies, it is important to remember that essential nutrients are cofactors for enzymes that are involved in the production of neurotransmitters such as serotonin and dopamine. Deficiencies in essential nutrients can lead to disruptions in neurotransmitter function, which can contribute to the development of depression. As Dr. Loftus's case study has demonstrated, deficiency in vitamin B12 can lead to depressive symptoms and other symptoms such as fatigue and weakness, which can mimic depression. In addition, the medical literature cites many instances in which patients had a major affective disorder and concomitant vitamin B12 deficiency, which made treatment of the patient's depression far more difficult and more complicated to treat. Folic acid, also known as vitamin B9, which many physicians recognize is involved in developmental neurobiology, is also important for neurotransmitter regulation, and when its levels are low, it can lead to depressive symptoms. There has been so much discussion about vitamin D lately in the medical literature and in popular media that I think everyone in this audience is probably aware that inadequate vitamin D levels have been associated with mood disorders. The good news is that addressing this deficiency can sometimes have dramatic clinical effects and can alleviate depressive symptoms. It seems that everywhere we turn, we are being encouraged to watch our intake of omega-3 fatty acids. There is a good reason for this because insufficient intake of omega-3 fatty acids found in fish and some plant-based sources has been linked to an increased risk of depression. As any biochemist knows, there are countless enzymatic reactions in the body that require magnesium. Perhaps, therefore, it is not surprising that magnesium deficiency can affect brain function and mood regulation, possibly leading to overt depression. It's important to note that while these nutrient deficiencies can contribute to or exacerbate depression, they are usually not the sole source of depression, although they can be. Addressing nutrient deficiencies through diet, supplementation, or medical intervention can be one aspect of a comprehensive approach to managing depression, especially in cases where these deficiencies are present. Another very important factor in understanding depression is being able to address questions such as what can we learn from single nucleotide polymorphisms associated with depression, or in other words, which somatic mutations can cause depression. One important hint is that Genome Wide Association Studies implicate various single nucleotide polymorphisms as being linked to deficiencies in essential nutrients and at the same time depression. For instance, the gene MTHFR encodes the enzyme methylene tetrahydrofolate reductase, which uses B12 as a cofactor, and single nucleotide polymorphisms involving this gene are associated with depression. A second example is the MUT gene, which encodes the enzyme methylmalonyl-CoA mutase, which also encodes an enzyme that uses vitamin B12 as a cofactor. Single nucleotide polymorphisms involving the MUT gene are associated with depression. Further examples come from the FAD2 gene related to omega-3 fatty acids, the GC gene related to vitamin D, and TRPM7 gene related to magnesium utilization, all of which are associated with depression for certain single nucleotide polymorphisms. I think that most physicians are familiar with SNPs, which are single nucleotide polymorphisms, but they have perhaps less familiarity with the field of somatic mutation, which really got started with the advent of the next generation sequencing. Somatic mutation is the gradual accumulation of mutation in the DNA of somatic cells in the body, not germ cells, that occurs over the lifetime of an individual. Somatic mutations can be point mutations, just single nucleotide changes, or can involve much larger segments of DNA, including deletions, insertions, frame shifts, mutations, etc. Causes of somatic mutations include errors in DNA replication and exogenous damage to the genome, which I will talk about in a bit in my presentation. Various consequences of somatic mutation are possible, including lethal somatic mutations, silent mutations, and somatic mutations that can lead to cancer. Genome Wide Association Studies have identified SNPs in these genes, listed here, that are associated with an increased risk of B-cell lymphoma and other cancers. This suggests that these genes may play a role in the development of cancer through their involvement in somatic hypermutation. Some of these genes, such as MTHFR and the MUT gene, can also be involved in other important cellular processes, such as DNA repair and methylation. Additional genome-wide association studies have identified genes linked to depression that are involved as key regulators of the stress response and in the metabolic turnover of catecholamines such as dopamine and norepinephrine. So SNPs in the gene COMT, for example, have been associated with depression, anxiety, and substance abuse. Additional genome-wide association studies have found genes involved in the timekeeping of metabolism at the cellular level, which are relevant for a relationship between depression and the aging process. There is a biochemical link between the process of aging and depression that is worthwhile to mention. The loss of DNA methylation over time due to aging process leads to dysregulation of gene expression that causes cellular stress and excess production of reactive oxygen species. An easy way to look at this has to do with the production of biochemical molecules by one cell that are needed as substrates for another cell. When this coordination of products and reagents gets temporarily disrupted for any reason, it causes problems. So it is becoming increasingly clear that there is a link between metabolism and somatic mutation. If that is the case, it raises the following question: might some of the products of metabolism mutate the human genome? In essence, it is a chicken or egg dilemma. But it might be even worse than a chicken or egg dilemma, because it might be the case that as some metabolic byproducts build up, they cause further somatic mutations, which causes further buildup of undesired metabolic byproducts. It can become a vicious cycle. How? We can ask what causes somatic mutation, and reactive oxygen species stand out as the prime candidate. In fact, reactive oxygen species serve a timekeeping function as switches of various cellular processes and physiological rhythms. The best candidates for timekeeping functions are reactive oxygen species oscillations, such as in hydroxyl radicals, which are produced as byproducts of bioenergetic metabolism. To counteract excess reactive oxygen species, cells contain antioxidant systems including peroxyredoxin enzymes, which protect the cell against oxidative stress. Mutants with defective peroxyredoxin enzymes exhibit increased somatic mutations caused by ROS. More about hypermutation as a consequence of gene activity during the oxidative phase of the redox cycle is shown on the next slide. For this, we go to our model system, yeast Saccharomyces cerevisiae, because yeast provide a wonderful example of how the redox system is cyclical. Even though yeast don't have lungs, they consume oxygen in a cyclical manner, as shown in the top left panel, much like breathing. Redox cycle generates an oscillating pattern of gene expressions, as shown in the bottom left panel. And another way to say this is that some genes are expressed in the oxidative phase, and different genes are expressed in the reductive phase. The right graph shows that genes transcribed in the oxidative phase accumulate mutations significantly more than genes transcribed in the reductive phase. The mutation frequency is plotted on the y-axis. Therefore, environmental stress that produces reactive oxygen species, such as spaceflight that we study at NASA, have the potential to mutate DNA and disrupt biorhythms. One of the most interesting points about this data is that reactive oxygen species induced by the environment enable a coupling between heredity and environment. This is why studying life processes in space provides an outstanding opportunity to unravel the timekeeping functions and somatic mutation at a fundamental level. In comparison to the known mutations that accumulate over the lifetime of an organism, the International Space Station induced mutation rate is 2 orders of magnitude higher than on Earth in every tissue examined in mice that spend less than 40 days on the ISS. Additionally, the genes that are expressed the most exhibited the highest rate of somatic mutation. However, despite all this data, we're not sure if the mice were depressed when they returned to Earth from the ISS. So the ISS-induced mutations lead to low levels of genetic mosaicism in different tissues, and some of the non-synonymous mutations likely induced loss of functions in areas of physiology relevant to spaceflight risks. So I hope you enjoyed exploring that depression can be thought of as a metabolic condition with a genetic feedback loop and the causal link between depression and reactive oxygen species, which are hyperreactive molecules that can damage cells and tissues, and that excess reactive oxygen species production can lead to inflammation and cell death, which have been implicated in the development of depression. So to conclude, in summary, depression is a common and serious medical illness that affects how you feel, the way you think, and how you act. It can lead to a variety of emotional and physical problems and can decrease your ability to function at work and at home. Growing evidence suggests that essential nutrient deficiencies and reactive oxygen species may play a role in the development of depression. Essential nutrients are cofactors for enzymes that are involved in the production of neurotransmitters. Reactive oxygen species are unstable molecules that can damage cells and tissues, and excess reactive oxygen species can lead to inflammation and cell death, and inflammation and cell death have been implicated in the development of depression. So somatic mutation may also play a critical role in the development of depression via a feedback loop with the metabolic mechanism of ROS production. A number of Genome Wide Association studies have identified single nucleotide polymorphisms associated with essential nutrient deficiencies and ROS. These SNPs have been involved in the development of depression by increasing the risk of essential nutrient deficiencies and ROS production. A high rate of somatic mutation was also observed in mice that spent only 5 to 6 weeks in space aboard the International Space Station, which prompts us to speculate that astronauts may undergo accelerated somatic mutation as well, which may lead to an accelerated aging process and potentially cognitive effects that are hard to predict, but we look forward to studying in the future. and I hope to tell you about it next time we meet.